So we're going to really jump into the cardiovascular system by talking about one of the two types of cells that make up your heart. We have got cardiac pacemaker cells and cardiac muscle cells. And so in this video, as you can see, we're going to talk about cardiac pacemaker cells. Okay, and so we've got these two outcomes for this specific video. Notice that in these outcomes, we've got certain words bolded. So we're going to talk about cardiac muscle cells or cardiac pacemaker cells as opposed to cardiac muscle cells. So cardiac pacemaker cells, they're also known as autorhythmic cells, and you probably saw that term on the outcomes. Okay, they don't have things like actin and myosin, or they have very little amounts of actin and myosin, and that's because they're not trying to produce force. Their job is just to tell other cells to do their job. Their job is to send signals so that cardiac muscle cells have action potentials. They're not force producing. Okay, so they're a little bit different than our typical muscle cell that we've talked about previously. What's really interesting about these pacemaker cells is that they don't need to receive information or input from the central nervous system for them to do their job. And what I mean by that is they don't need to receive a signal from the central nervous system to cause them to have an action potential. They will absolutely respond to that, but if they don't get a signal for whatever reason, they'll still generate an action potential. And so you can look at this as one of those fail-safes that your body has in case something goes wrong. And so we didn't look at skeletal muscle cell action potentials, but you can see what it looks like on the right. Okay? And then now I want you to look at the cardiac pacemaker cell action potential on the left. And as you can see, they're very different. And one of the things that stands out is that there is no resting potential in a cardiac pacemaker cell. And we're going to talk about that in just a second. Okay, but notice that there is essentially three phases. We're going to talk about each of those phases individually starting now. Okay, and just here we've got this blown up view. All right, so we've got three phases. I'm going to highlight each of the phases as we go through this. Okay, now each of these phases has one ion that's responsible for it. Okay, now on the bottom here we have these permeability curves, and what I mean by these permeability curves is all down here. Okay, these are not, as they go up, it's not having anything to do with membrane potential. What it's telling us is that as the curve goes up, that means it's easier for that specific ion to enter or exit the cell. Okay, so for example, here at this point, it's really easy for sodium to move across the membrane. At this point, through specific channels, calcium, this is also calcium, and then over here is potassium. Okay, and when you know when the permeability is high, you'll know what phase or you'll know what ion is responsible for a specific phase. So during this drift phase, voltage-gated sodium channels are open, which allows for sodium influx. There's more sodium outside than inside. It goes down its gradient. And so membrane potential becomes less negative. Sometimes this is called like the pre-depolarization, okay, because the membrane potential is actually depolarizing. So why do we have this drift phase? Well, this is a slow depolarization, okay? And it's spontaneous, meaning there's not a stimulus needed for this to occur. And this is why pacemaker cells don't need input from the central nervous system, because they will undergo this slow depolarization regardless of whether or not the central nervous system is communicating with it, okay? Now, usually, in a perfect world, as you're sitting here, as you're doing whatever you're doing watching this video, the central nervous system is communicating with those pacemaker cells. But just imagine if for some reason that connection between your central nervous system and your heart was broken. If the pacemaker cells couldn't generate these spontaneous action potentials, that would probably result in death rather quickly. Okay? And so this allows pacemaker cells to continue to function even without the central nervous system. In other words, it's a fail-safe. Okay, so we're going back, we're at drift here. So the next phase after drift, once we hit threshold, is depolarization. Okay, now there are two different types of calcium channels that open. You'll not need to know that there are two types or what the difference is between these two types, but there are two types of voltage-gated channels, voltage-gated calcium channels that open during depolarization, and this allows for calcium to go inside the cell. Now, some of you might be thinking, oh, calcium, it's going to cause cross bridges and all that, because we're talking about cardiac cells, which are a type of muscle cells. In this case, calcium, that's not its job. Its job is just to depolarize, Remember, there's not really a lot of actin and myosin in pacemaker cells, so we're not trying to generate force. And then finally, we've got potassium channels. Now, these potassium channels open, or in other words, they're really open during repolarization, which allows potassium to leave the cell, so we have a repolarization. All right. Now, unlike a neuron action potential, there's no hyperpolarization here. So once repolarization ends, we go back to that drift. It's important to remember all of these are voltage-gated channels. I'm not going to ask you to remember specific values of when these open and close, but just know that each time we go from one phase to the other, we've got a set of voltage-gated channels that's opening and essentially a set of voltage-gated channels that's closing. So for example, when repolarization starts, calcium channels are closing, potassium channels are opening. When repolarization ends, potassium channels are closing, sodium channels are opening. Okay? And we can look at this all together, and then, like I said, it's really messy, but you can see where different ions are more permeable than others. So drift is caused by sodium influx, depolarization is caused by calcium influx, and repolarization is caused by potassium efflux. And that's all we need to know for now about cardiac pacemaker cells. So can you do these three things? Can you draw out a cardiac pacemaker cell action potential? Can you identify the ions responsible for each phase? And can you tell me why we have a drift phase?